would now like to introduce your first presenter for today, Erin Tesh, Senior Vice President and Managing Director. Erin, you now have the floor.
Everyone. I'd like to welcome you all today to the webinar. Good morning and good afternoon, wherever you're located. I'm Erin Tesh. I'm Managing Director of TSG, and I'd like to start off by saying thank you for joining today. I hope all of you are well and your families are staying safe and healthy during these challenging times. I'd like to introduce my colleague, Dr. Leslie Patton, who will be presenting today. Leslie is a senior consultant at TSG and has more than a decade of experience in regulatory toxicology. Leslie specializes in food additives and food contact substances, pesticides, consumer products, and cosmetics. Leslie routinely prepares the necessary submissions for food contact notifications and GRAS notifications. Leslie has a Bachelor of Science from the University of Michigan and a PhD in environmental toxicology from Cornell University. I recognize none of you really signed up to hear me speak today, so I'm going to let Leslie begin here soon, but I wanted to just spend a few minutes for those of you that are not familiar with TSG Consulting to just give you a few words about our firm. So please bear with me for a few minutes. TSG has been providing companies around the world with regulatory guidance and scientific expertise for over 30 years. We help clients address various technical and regulatory issues associated with taking their products to market in multiple jurisdictions. We have offices in the U.S., Canada, and throughout Europe, and we have consulting partnerships throughout the world. We are part of Science Group, which is listed on the London Stock Exchange. TSG is one of 6 companies under Science Group. We serve a number of key markets and industry sectors, including agricultural, industrial, consumer, food and beverage, animal health, and medical. Our teams are comprised of scientists and regulatory experts, many of whom have previously held positions at regulatory agencies such as FDA, EPA, CPSC, and USDA. We have many staff members that have also come from industry. Science Group has over 500 staff members. We speak over 30 languages, have 12 offices around the world, and 2 dedicated facilities. I do encourage you to submit your questions throughout the presentation via the chat function. I will be monitoring the questions, and I'll do my best to get to all the questions. We'll have about 15 minutes at the end of the presentation to answer your questions. Also, as most of us now are navigating the world through our home offices, please recognize that Leslie and I are actually both at our home offices. We think we've worked out the kinks here, but please be patient with us. So with that, I'll turn this over to Leslie.
Hi, thanks, Erin. Thank you, everybody, for coming. Today I'm going to discuss, give you an overview of food contact substance regulation in the U.S. With a special focus on antimicrobial jurisdictions. And then I'm going to talk about some options for food contact substance approval, with a particular focus on FCNs, or food contact notifications. I will discuss some challenges that we see when we submit notifications, and then provide some tips for successful submission. So what is a food contact substance? The FDA defines an FCS, a food contact substance, as any substance that is intended for use as a component of materials used in manufacturing, packing, packaging, transporting, or holding food, if such use of the substance is not intended to have a technical effect in such food. So things that are considered FCSs include polymers that comprise food packaging, any additives or adjuvants that are added to those polymers. Certain antimicrobial substances are considered food contact substances. Also, what FDA terms secondary direct additives, which are substances that have a technical effect in food during its processing, but not in the finished food product. Those are also FCSs. Some substances that have been in use since before 1958 are called prior sanctioned substances by FDA. Those are listed in the Code of Federal Regulations under Section 181. And then also some GRAS substances generally recognized as safe for their use in food contact. The regulation of food contact substances started in 1938 with the Federal Food, Drug, and Cosmetic Act. This was the act that gave FDA the authority to regulate food additives, and that included food contact substances. The act was amended in 1958 to establish a pre-market approval process for food additives. And more recently, in 1997, the Food, Drug, and Modernization Act established the Food Contact Notification, or FCN, system. There are other legal, there are other laws that govern food contact substances, including some that are administered by USDA, as well as EPA, in particular with respect to antimicrobials. The antimicrobial legal framework requires its own slide because it's pretty complicated. In 1996, the Food Quality Protection Act changed some of the regulatory authority of FDA. Previously, FDA had jurisdiction over antimicrobials used on processed food and in food packaging material. The FQPA gave authority over antimicrobials for those uses to the EPA. There was a bit of grumbling about that, and a couple years later, Congress enacted the Antimicrobial Regulation Technical Corrections Act, or ARTCA. And this created a situation where antimicrobial residues require clearance by FDA as food additives under Section 409 of the FDCA, and also are regulated by EPA under Section 408 of FDCA, and under another law that that EPA administers called FIFRA, the Federal Insecticide, Fungicide, and Rodenticide Act, and that's the law that governs pesticide registration and defines the term pesticide. So this made all for a bit of a mess with antimicrobials registrations or regulations. And so FDA has published this chart on its website in an attempt to clarify the jurisdictional difference or division between certain antimicrobial uses. So if you go along the top, you can see that in general, food that is processed or that is going to be processed is considered, those antimicrobial substances are considered food additives and are regulated by FDA. Also in processed water that is present in a food processing facility. If the substance, if the antimicrobial is controlling microbes on a raw agricultural commodity, so this is like a fruit or vegetable out in the field, then it's regulated as a pesticide by the EPA. If you look at the bottom, I'm going to go into a little more detail in the next slide about that if it's not clear. If you look along the bottom, you can see that EPA gained control of surface sanitizing solutions, things like spraying countertops, and then also equipment in food processing plants where the surface needs to be antimicrobial. Those things are regulated by EPA. FDA in general has control over antimicrobials applied to food packaging and antimicrobials where the antimicrobial effect is not on the surface of the food contact article, or if there's no antimicrobial effect in the finished article. And I'll explain this a bit more in a moment. You will see here that some of these FDA regulated uses, next to them it says may also be subject to FIFRA. Generally that means that a product must be registered as a pesticide even though it is regulated under FDCA as a food additive. So it may be simplest to think of this in terms of what is regulated as a food additive by the FDA only. So FDA regulates anything added directly to food. So a food preservative, for example, sodium benzoate, is not under EPA's purview. Antimicrobials used in poultry and meat processing plants are not subject to EPA regulations generally. They are subject to certain USDA regulations. And then also antimicrobials used on post-harvest seafood are regulated by the FDA. The FDA regulates antimicrobials used in water that delivers antimicrobials to food during processing, but not if the purpose is to disinfect the water itself. And then finally, the FDA controls or regulates substances that are supposed to control microbes on raw agriculture commodities if they are then being processed or transported to a processing facility. And FDA in ARCTA has defined, or the law defines, what does processing mean. And I've included a bunch of verbs down here that FDA and EPA have agreed, equate to processing of food, and also other verbs that do not equate to processing food. So you kind of need to use that guide sometimes when you're trying to decide who's going to regulate your antimicrobial product. So types of products that are regulated as food additives under Section 409 of the Food, Drug, and Cosmetic Act, and not meeting the definition of pesticide chemical under Section 408 of that act, they still require registration as a pesticide under FIFRA. And these include things, so I put this first one is what I included on the last slide, just to be controversial. Treatment of raw agricultural commodities during processing, that's an FDA use, but it would behoove you to ask EPA if it is also controlling a pest on the raw agricultural commodity, because if it is, then it would be considered a pesticide. An antimicrobial applied in or on food packaging. Again, this is considered a food additive and regulated by FDA, but it may also need to be registered as a pesticide. Antimicrobials used in contact with food that have no intended ongoing effect on the final article or on the article's food contact surface. So this would be something like a preservative that's in a colorant, and the colorant is going to be used to make a food surface countertop, say. So the purpose of the antimicrobial is to preserve the colorant, not to have an ongoing effect on the food surface countertop. This also falls in the category of a dual EPA-FDA jurisdiction product. And then finally, an antimicrobial that is intended to have an ongoing effect on any part of the object except the surface. And this is something EPA and FDA call these material preservatives. The antimicrobial only preserves the article itself. It doesn't kill microbes on the final article. So it only preserves the materials in the article. So this is, for example, an antimicrobial that's embedded in a resin that you then make, and you make that resin into a food contact substance. So now that dual jurisdiction is clear as mud, let's move on to what are your options for a food contact substance approval. So the main one I'm going to talk about today, and probably the most popular one, is the food contact notification. You can also submit a food additive petition. You can submit something called a threshold of regulation or TOR request. You can determine that the substance is GRAS for this purpose. And finally, you can determine that the substance is not likely to be incorporated into food and is therefore not a food additive or food contact substance. So food contact notifications. This is a program that started with the Food and Drug Modernization Act. It was launched in the year 2000, and as of this morning, I checked. I think there's 2,079 effective food contact notifications that have been established since 2000. The type of a substance that is appropriate for an FCN is, as FDA defines any food contact substance, is a material used in manufacturing, packing, packaging, transporting, or holding food. So these are, FDA calls what they call indirect food additives, polymers, monomers, polymerization aids, adjuvants, any equipment components of food processing equipment or radiated packaging. Also secondary direct additives, which are substances that have a technical effect during the processing of food, These are things like boiler water additives, processing aids, and ion exchange resins. So what goes into a food contact notification? The first, the chemistry section is the longest and most data intensive. You need to clearly identify the substance. So that includes not only the name and the trade name, the CAS number, that type of thing, but you also have to you have to provide spectra, IR, NMR, MS, that kind of thing. And if it's a polymer, you need to characterize the distribution of molecular weights and the fraction of low weight oligomers. The analytical methods when you're characterizing your substance in the laboratory, or if you do a migration test, and we'll talk about that a little more later. But anytime you provide FDA with analytical methods, they have to be very well described. You need to include details on accuracy, precision, selectivity, the level of quantitation and level of detection, such that the FDA's chemists can follow your description. You need to include standard curves at relevant concentrations. And you also have to fully validate the method and provide the FDA with that information. So things like you need to do replicate analyses of fortified analytes in the matrix, in the appropriate matrix. You need to determine the percent recovery of a fortified analyte. And you also need to demonstrate that the analyte, which is a food contact substance, is stable under the conditions of testing. The chemistry information should include physical and chemical specifications such as melting points, density, solubility, etc. And then also a description of the manufacturing process is necessary. And this is a section that sometimes trips up notifiers. It has to be very detailed. You need to include a list of your raw materials, including any catalysts, any chemical reaction that takes place during the manufacture of the substance, the reaction conditions, purification steps, and then you have to identify impurities and potential byproducts, anything that may potentially migrate into food. Number 6 here, the use level and the pattern. So you need to decide what is the use level of your substance in the final article that will be sold. So you have to define that in the food contact notification, and then the pattern of use, and that means what type of food will it contact, under what conditions of temperature will it be exposed to during its typical use. The intended technical effect needs to be demonstrated. So you need to show basically that the substance is doing what you say it's going to do. So, for example, if it's antimicrobial, you need to have data showing that it has antimicrobial effects at the use level and under the conditions of use that you are that you will be using it for. You also have to show that you're not using too much of the substance. So you need to show FDA that at a lower level it's not efficacious, because FDA doesn't want you to be using more than is necessary to get the job done. So you have to provide that type of information in the submission. And then the last part of the chemistry section is you provide information about how much of this substance may migrate from a food contact material into food. And in order to do this, the most obvious answer, I think, is to carry out a laboratory migration study. The design of the study will depend on the intended use of the food contact substance. So the conditions of temperature, the food simulants that you use will all depend on what your intended use is. The migration studies give you nice, solid information. Of course, they're they're expensive in both on resources, and so it's always good to start before you launch into a migration study in a lab to model migration. And this can be done in a simple way. So if you assume that 100% of your food contact substance migrates from packaging into food, if you can show that that level is safe, then you don't need to go and do a laboratory test to characterize migration any further. And FDA, in its guidance, has some default assumptions you use when you model migration. For example, they assume that 10 grams of food is in contact with 1 square inch of packaging. There are some other models that we use to estimate migration that are a little bit more sophisticated than a mass balance model, and they rely on how much does the compound diffuse through certain kinds of polymer, and how do the conditions of temperature affect that. And so using these models, you can often get a good picture of migration, a good enough picture of migration that you can say, well, this level of migration is safe in food. And if you can't, then you would move on to doing a laboratory migration study. So the next part of this is the exposure. How much of this substance is going to make its way into the diet? Before you can estimate exposure from migration, you need to consider the types of food contacting the food contact substance, and also the packaging type that your substances will be in. So the FDA publishes something called food type distribution factors, which is in this equation here, these little F's. These are the fraction of all food contacting each type of packaging material that is aqueous, acidic, alcoholic, or fatty. And what it does is it takes your migration calculation, the calculation of your total migration, and it scales it so that you get the migration value from each stimulant or each food type according to the actual fraction of food of each type that will contact the article. The dietary concentration can then be calculated once you calculate this migration amount. The dietary concentration can be calculated as the total migration times a consumption factor. The consumption factor is the fraction of the daily diet that is expected to contact a certain specific packaging material type. And these are provided, default consumption factors are provided by the FDA. They're in FDA's guidance. So, for example, they assume that 80% of the diet is in contact with some type of polymer, and that 20% is in contact with some type of paper. And then those consumption factors are further refined, by a specific type of polymer, a specific, a paper, a coated paper, a non-coated paper, that kind of thing. There's also consumption factors for adhesives, glass, a lot of different substances that contact food. Now you can calculate your own consumption factor, because sometimes the default values are higher than you think your substance is actually. If you have information that can modify the consumption factor and show that the fraction of the daily diet that's expected to contact your substance is lower, then if you provide that to FDA, they may accept your consumption factor. The estimated daily intake is calculated assuming that every person eats 3 kilograms of food a day. So that's the dietary concentration times 3 is the estimated daily intake. And then you need to figure out what the cumulative EDI is. And the cumulative estimated daily intake takes into account other regulated uses of this food contact substance. So, for example, if there's a food additive petition on the substance, or if there's other FCNs for your same food contact substance, you would need to add those exposures to get the cumulative exposure, and that's important. Oh, and I should note that if the FDA can provide you with that number, that cumulative number. The cumulative number is important because the cumulative exposure determines how much toxicology data that you need to provide the agency. So the toxicology data requirements are tiered according to the level of exposure. And so if you determine that the exposure is equal to or less than 0.5 ppb, I'm sorry that this number didn't make it onto the slide. I apologize for that. But if it's so low that's less than 0.5 or equal to 0.5, then you don't need to provide any toxicology data. You just need a brief discussion of the substance's toxicity and its carcinogenicity, if that's relevant. For dietary exposures between 0.5 and 50 parts per billion, you need some genetic toxicology data, an Ames test, which is a test in bacteria, and then an in vitro cytogenetic test in mammalian cells or a mouse lymphoma assay. Hopefully those are both negative, meaning they're not genotoxic. If they're positive, you'll need to do a little bit more work and submit some more data. For dietary exposures that are greater than 50 parts per billion but less than 1 part per million, you need those same genotox tests that I just mentioned, and also an additional in vivo genotox test, a micronucleus test. And you also need 2 90-day subchronic studies, a rodent study and a non-rodent study. So, as you can see, this gets a little bit more expensive. If your dietary exposure is greater than or equal to or greater than 1 part per million, you may need to submit a food additive petition rather than a food contact notification. And this would require a lot more sort of intensive data, chronic study, reproductive toxicity study. If an ADI already exists, and the ADI is the acceptable daily intake, so that's sort of a reference dose that's safe, let's say. So if an ADI already exists, you may need a bit less data. If an ADI exists for a similar chemical to your food contact substance, you may be able to bridge those data if you can, if you have some information about the toxicity and the metabolism of the food contact substance. So all of the studies should follow FDA's Red Book. This is something that's available online, and it provides test guidelines. The other thing I should mention is that these studies don't all have to be done if the data already exists. So if you have a substance that's a common substance that's been used in food or in anything else before, and these studies, these genotox and tox studies already exist, for example, in the literature or in other FCNs, you can reference that literature and those FCNs in your submission. And it may preclude you from having to do these tests yourselves. And so the toxicity profile you do that to inform this overall safety assessment, which is kind of your summary of showing FDA, here's my exposure amount, here's the safe amount, and this is why this food contact substance, as I'm proposing it be used, is safe. The last section of the FCN is the environmental assessment. For certain uses, the environmental assessment is not necessary. If you look at the Code of Federal Regulations under Section 25.32, there's a list of what they call categorical exclusions that exclude you from having to do this environmental assessment. And those are things like if the substance is less than 5% by weight of packaging material, or if it's a coating, or if the substance is already regulated by EPA as a pesticide, then you may not have to do the environmental assessment. If you do have to do it, it includes a description of the proposed action. So the proposed action is, again, your intended use, what you're making, a discussion of some alternatives to that action, and this includes, and then an evaluation of the impact of the action. So things like you have to talk about the introduction of the food contact substance into the environment from manufacturing, from its use, and from its disposal. You need to discuss the environmental fate of the substance, once it is in the environment, in water, usually it's in water, it could be in air, it could be on land. And then you need to discuss it in ecological toxicology. So assuming, you calculate your environmental concentration that you produce as a result of manufacturing the substance or the use of the substance, you need to show that it's not going to be toxic to fish or other wildlife. If you do an environmental assessment and it's accepted, it will be published on FDA's website. It is a public part of the FCN. Now, it can have confidential parts, and what you can do is submit 2 versions of it to FDA. So FDA can publish one of them on its website and keep one of them confidential. So what exactly happens when you submit an FCN to FDA? Well, first of all, you submit it to the Center for Food Safety and Applied Nutrition. That's the division of FDA that this is going to. It's also known as CIFSAN. They have the FDA has a pre-acceptance kind of screening procedure to look for any major deficiencies that may affect the acceptability of the submission. And that usually happens within, I wrote 3 weeks here, but it has definitely taken longer at certain times. So it takes place within about a month. And then you receive an acknowledgement letter after that time saying that it's passed phase 1 of the review process and that it's entering phase 2. Phase 2 is a more detailed review of all the data that have been submitted. They look at especially your dietary exposure. They look at it with respect to other FDA-regulated substances. They do a carcinogenic risk assessment if that's relevant, and then they they have 120 days to do this, and this is written into the law. So technically, if you don't hear back from FDA after the 120 or by the 120th day, you can start selling your food contact substance. Usually that's not what happens. Usually there's something that needs clarification or some perhaps deficiency, and they can be small or large. And you'll get a letter, and FDA details what you need to do at that point. Most deficiencies can be addressed in 10 days, which is the amount of time you have, 10 business days. And so you reply to FDA's concerns and answer their questions. Sometimes there is a deficiency that is, FDA is asking for more testing or something that can't be accomplished in 10 days. And so for something like that, you would just withdraw your FCN. And you withdraw it without prejudice. There's no penalty for doing this. But assuming that everything's fine, you go forward, you submit your response within 10 days, then they'll hopefully accept it. And I should also mention depending on the kind of how big the issues are in the deficiency letter, that 120-day clock can technically restart. They don't always do that, especially if there's just some more minor deficiencies. But it does buy them more time, so you should keep that in mind. That 120 days doesn't always equal 120 days. But anyway, at the end of this, the FCN is published on the FDA's website. There's an inventory of effective FCNs, and that usually happens within about a month of them accepting it. It makes its way onto their website. So that's all I'm going to say about FCNs for now. If your food contact substance has a technical effect in food, or if it's migrating into food at a very high concentration, as I mentioned before, if it's at least 1 part per million or more, then you may need a food additives petition instead of a food contact notification. This requires rulemaking, and so eventually once you submit it and it's successful, it'll be listed in the CFR along with any specifications and limitations. It's a similar process in that you collect all your data. There's more data though. There's all of those additional toxicology studies. You submit that to the agency. There's probably some back and forth with agency asking for more or asking for clarification and that kind of thing. And then once they accept your submission, federal rulemaking starts, And there's, so there's a comment process where there's a draft published, and then eventually a final regulation is published after the petition is accepted and the draft is accepted. So the whole process is long. I say here 2 years minimum, but that's really for the rulemaking. I mean, the 2 years is probably closer to 2 to 5 years. Let's say that. It's a long process. It's data intensive. If you can avoid a food additive petition, that's better. Or sometimes, if you're making something that has a technical effect in food, then that's the route you have to go. Another way of selling a food contact substance is to determine that it's GRAS for that purpose. So I'm not going to say a lot about GRAS. This is an area that I do work in, and so if you have questions about it, feel free to contact me. I'm just not going to talk about it a lot here. There is a list of substances that are GRAS for food contact listed in 21 CFR Section 186, and then there's also an online inventory of GRAS substances on FDA's website. A threshold of regulation exemption is another submission that you can do to FDA. It's a process that preceded the FCN process. These are, the general idea behind these is that they do not enter the diet at very high concentrations, and so therefore they're considered below the need for regulation. And that dietary exposure threshold is 0.5 parts per billion. So if you can show, with migration testing and or calculations that the dietary exposure is below 0.5 parts per billion, you may be able to submit a successful TOR exemption. The substance also has to have no structural basis for toxicological concern. The good part of TOR submissions is that less information is required compared to an FCN, or certainly a food additive petition. For example, you don't need to describe your manufacturing process in these. So there's definitely less labor intensive from that point of view. On the other hand, there's no 120-day review period for TOR exemptions. So there's not really any certainty regarding how long it's going to take FDA to review your submission. So that's one downside. Another downside is that the approval of the TOR is not limited to the submitter. That's not the case for an FCN, and I didn't mention this when I was talking about FCNs, but your FCN is only for you. Your competitor can't use your FCN to sell their own version of that food contact substance. That's not true of a TOR exemption. Once that's published on FDA's website, anybody can use it. And then the last thing I'm going to talk about for food contact substance is an opinion letter. And this is something, it's sort of related to the TOR exemption, except you're not making a formal submission to FDA. But it's basically a letter typically written by an attorney, and the letter concludes that the substance is below the threshold of regulation, and therefore can be considered not to have migrated into food and won't become a part of the diet. So again, with most substances, the threshold here is 0.5 parts per billion in the diet. If the substance is toxicologically problematic, the level will be lower than that, and that's what's called a no migration determination. The basis of the letter can also be something called a functional barrier, which you see in packaging sometimes. There's a barrier in between the food contact substance and the food itself that is known to prevent the migration. So that's another way to sort of avoid regulation, if you will, is through an opinion letter. All right, so some of the challenges that I've seen in food contact notification submissions, probably the biggest one has to do with the analytical chemistry data, both the characterization of the substance and any analytical chemistry that's done in a migration test. As I noted before, the validation process is very important, and FDA details what they want to see in their guidance document. And if you don't have that document, I can, contact me and I can send it to you, or you can probably find it just by Googling SCN chemistry guidance for SCNs. But that you need to follow FDA's recommendations for validation. Another part of the FCN that tends to trip up people is the manufacturing process description. And that's just because it needs to be very detailed. The FDA chemists want to be able to basically create the substance themselves. That's how detailed it has to be. And so that's something that we often, with our clients, go back and forth on a lot, sort of, getting the information out of them. And then carrying out a migration test can be challenging, sort of for some of the reasons I noted above, the analytical chemistry, but also setting up the test and getting the conditions right. You need to be sure about what your intended use is, because the migration test reflects that use. So you need to make sure you know what you want at the end of this process, before you set up your migration test. And here are some tips for submission. The first one here is have a pre-notification consultation, or what they call a PNC. This is really important for if you have a substance that's novel, that is not yet regulated by FDA. Then that's when I would go get a PNC. If you have some data already, some chemistry data characterizing your substance, or really anything, they will review those data and provide you with feedback. They're very helpful. They do want to help you. They don't want to make more work for you or for themselves by having you submit something that's not complete. So I'm a big fan of PNCs, and they can all be done on the phone, or even through email, sometimes. So it's not something where you have to be there in person, although it can be. Although probably not right now because of coronavirus, but in normal times you can go and meet with them in person if you want. You should always, as I noted, attempt to model migration before investing in a laboratory migration study. Sometimes you find that your model gives you the answer that you need. In other words, your model tells you that a low level of substance is migrating, a level that's not of concern. And so then you don't have to do the study. Even though you're just estimating the migration concentration, it's good enough if your calculations are sound. Establish a dietary concentration. Estimate the dietary concentration early on. And the reason for this is because that dictates how much toxicology data that you need. So it's good to know that from an early stage. What's the estimate of the dietary concentration? And again, I think now this is the third time I've said this, but provide FDA with sufficiently detailed manufacturing process information such that they could make it themselves. You need to characterize not only the substance, but also the impurities and byproducts. FDA wants to know if those will make their way into the diet as well. And if they can potentially make their way to the diet, then you need to detail their toxicity in addition to the toxicity of the food contact substance. And then finally, I wrote here, use FDA's database of environmental assessments. These are really handy because FDA actually encourages you to use words. I don't want to use the word plagiarize, but they encourage people to use the same words that previous environmental assessments have used. They don't want you to sort of reinvent the wheel on these. And so if you can go through the database and find a substance that is a similar intended use to yours, then you can use that existing environmental assessment and modify it as you need to for your substance, but definitely don't start from scratch on these. So how can TSG help you? Well, we can help you with your reviewing the analytical data that you collect, with your method description, your method validation. We have a lot of experience creating migration models, and we use some computer models to model migration, calculating and estimating dietary concentrations. and then also just overall, the SCN is just one form, but with all the data that are required to go with it, you end up attaching 30 or 40 attachments to this one form, and FDA has very specific ideas about how those should be attached and what they should be named and that kind of thing. And it's easy if you've done it before, but it's a little bit of a nightmare if you never have. We also have good relationships with the consumer safety officers at FDA, and we can communicate with them along the way before the submission and after the submission. And with that, I'd like to conclude my talk by mentioning that we have a white paper that we've written. It's called Polymer Colorants and Antimicrobial Food Additives, a look at the complexities of food contact notifications. And we will email you this paper along with a copy of this slide deck, probably sometime next week, as a thanks to for joining us today. And with that, I will conclude my my webinar, and I'm interested in your questions. And thank you for listening.
Thanks, Leslie. We appreciate it. That was really informative, and certainly you painted the picture of the complexities associated with the process, particularly the EPA versus FDA jurisdictional issues associated with antimicrobial additives. I know we get those questions a lot, and quite frankly, I think some of the staff within each agency are not entirely sure of the jurisdictional responsibilities as well. We did get some questions. We have a few minutes to go through the questions. So let's get started. One of the questions is if a given substance is duly regulated by FDA and EPA, does it require both FIFRA registration and FDA clearance? That's number one. And is there any type of EPA approval other than FIFRA registration?
So, yes, it may require both. And what you would need to do is you would need to do an FCN first and get that done, because you need, in order to register the pesticide with the label that you want, with the FDA uses on it, that requires you to submit the accepted FCN or proof that you have to have the FCN accepted. Okay. And then the other part of the question, so some food additives need to be registered as pesticides, and some food additives need to have, that are also pesticides, and need an EPA risk assessment. And the risk assessment at EPA is it uses different assumptions, and it's actually a little bit more stringent, frankly, than FDA's. And so that's sort of the other piece of that the EPA may need to have a say in for these duly regulated products.
Okay, good. All right, thank you. Here's I'm not sure we're going to be able to get to everybody's questions, but I'll do the best we can. If you withdraw an FCN, is it allowed to perform additional tox testing and then submit for the same substance for a second time?
Yes, absolutely. I mean, there's no penalty for doing that other than, you have to start over in that 120-day process. Okay.
So, do FCNs require electronic-only submission, or is it paper?
No, you can do either. So, there is now a portal that you can submit all of this into FDA, and they encourage people to do that. It is I can't think of the name of it right now. Some acronym, but I find it works well sometimes and not at other times. I'm not a huge fan yet, but we do it that way. But you can still submit an SCN as a paper submission, or I'm calling it paper, but it's actually, I will put them on a disc, which seems old-fashioned now, but you put them on a disc and send them in the mail to the agency. They will accept either type of submission.
Got it. Okay. So does an FCN, does FDA require a U.S. agent or can foreign companies? Okay.
No.
Okay. Can you modify an FCN?
Yeah, so depending on how big of a modification it is, if it's a minor modification, and there's a whole FDA document that describes what is a minor modification versus not minor. If it's minor, there's a different form. You can file, there's a specific FDA form that you can file that's a modification of an existing FCN. If it's a major modification, though, you may just need a brand new FCN.
Okay. And can you transfer an FCN from one company to another?
No, well, so here's what if your company changes names or changes hands, then you can all you have to do is write FDA a letter telling them that, so now your FCN is for your new company. But you can't like go out of business and then give your FCN to someone else or something like that. You can't change who the manufacturer or who the notifier is.
Mm-hmm. Okay. So have you found that, given COVID, that you've seen greater delays of FCNs, or has that affected the process at all?
Yes, it has. I mean, it's affecting FDA in general. The good thing again about FCNs, though, is that this 120-day clock is written into the law, and so they still have to review your FCN in 120 days. So that piece of it is not any longer. Other parts of it, communicating with FDA is a little bit more challenging, I'd say.
Mm-hmm. Okay. Another question we have here is, let's see, do the safety studies submitted to FDA have to be done under GLP?
Yes.
Okay. And what needs to be included in a pre-consultation meeting? I mean, what kind of information do you need to provide in order to get a meeting?
It depends on where you are in your process, but the best. So all you do to get the meeting, all you do is email them and ask, and you ask for it. You describe what you're making and what your questions will be, and then I think, though, the best way to get information and the most help you can get out of them is to submit all of the data you already have. So if you've done some analytical testing already, if you've done some migration studies, submit it all to the agency with the PNC, and they will review it and provide feedback to you. If you're not at that stage, though, if you're really early in the stage and you just need help at that point, they will also help you at that point. Okay,
terrific. Well, unfortunately, we're not able to answer any more questions because we're at the top of the hour here. But I'd like to thank you all for attending. As Leslie said, we will be providing you with a copy of the slides as well as the paper the white paper that Leslie wrote. And please have a great day, and please don't hesitate to reach out to us if we can be of help. Terrific. Well, thanks so much, everyone.
Thank you. Bye-bye.
Bye-bye.